All right, so this is chapter 20, and this is the core material, so this is for everybody. So what this is DNA technology and genomics. So it's kind of looking at the idea of some of the techniques that biochemists and molecular biologists and stuff use to analyze DNA. So the main idea here is that biologists have developed techniques for artificial manipulation of DNA, cells, and organisms. Okay? So in your TOK portion of your notes, I need you to use to answer the following questions. It says the use of DNA for securing convictions in legal cases is well established, yet even universally accepted theories are overturned in light of new evidence in science. What criteria are necessary for assessing the reliability of evidence? So as always, take some time, write down an answer, and then at some point we'll go over these in class and I'll be checking them within your notes too and giving you some credit for them. Okay? All right. So some of the challenges to studying DNA is that it's very, very small. Obviously, if 6 feet of it fit within each one of our cells, then it's very tiny. Okay, it's just a thread-like macromolecule that's very difficult to see unless you have an electron microscope, of course. So naturally occurring DNA is really long. It only comprises a small portion of the actual DNA molecules is a gene, which is what contains the information that gives us our particular phenotypes and stuff. So there might only be a small part of the DNA that actually codes for a gene within that chromosome. Okay, and then there might only be some small differences on either side of this gene regarding the actual nucleotides themselves. Okay, so to overcome some of these challenges, scientists have developed a lot of different types of methods for gene cloning, for making more of the DNA, making it more visual, being able to do all of these different types of experiments with it. Okay, so what we do a lot of times in terms of cloning is we're going to make copies of the particular gene or the product so that we can get the actual protein out of it. And cloning can be used to give an organism a new type of metabolic activity, a new appearance, make it bigger, more desirable. Some of the examples are pest resistance, drought resistance, pesticide resistance, a whole variety of things. So what we're basically doing when we do that is we're going to take genes and we're going to insert them into another organism. And one of the examples that we'll go over here, and I'll show a video about it, deals with insulin and using E. coli to produce this insulin. Okay, and that was kind of one of the first things that happened in terms of this studying of the DNA and making these transgenic organisms so that we could produce insulin using the E. coli, and that kind of gave birth to this whole field of molecular biology. Okay, so most of the methods for cloning of the DNA share some general features. What we're typically going to do is that molecular biology rat, so to speak, the E. coli molecule, we're going to take advantage of the fact that they have plasmids, and we're going to isolate these plasmids from the bacteria. We're going to find a way to get the gene into it, and then what we have then is a recombinant type of a plasmid, something that contains DNA from 2 different sources. You have a bacterial cell, and that's going to then kind of get this, take up this plasmid in transformation, like we've talked about in the HL lecture. And so now what we'll do is we'll grow these bacteria, and when they reproduce, they're going to reproduce all of the stuff within them. So we're going to start to have a bunch of clones of these particular types of cells, ideally, given that no mutations have occurred. And then as this cell is reproducing, as these plasmids are reproducing, what we're doing is we're cloning the genes. Those things are getting copied at the same time. And then we can use this process for a variety of different types of techniques. So that's going to kind of be like the underlying aspect of what's going on here with molecular biology. So here's a video that I'll post underneath this lecture that I'm going to want you to watch. It'll be like the production of biodiesel, or it'll be titled something like that. Okay, so it does a good job of explaining how transformation works. All right, so basically what you're doing, what you can see from this particular picture here is we've got this bacterium, okay, and what we're going to do is we're going to take a gene out of a particular organism that we're interested in cloning, and we're going to insert it somehow into this plasmid, and then we're going to get this bacterium to take up that plasmid and become a recombinant bacterium. What that simply means is it's going to contain genetic information from 2 different organisms, and then what we're going to have to do is really select for somehow, and we'll kind of go through this in another one of the lectures coming up. We're going to have to select for the presence of this gene in the bacterium. And then what we can do is start to take, once we've identified that gene of interest, we can start putting it into particular organisms, and we can start producing the proteins that we're after. And then that will allow us to do a variety of things. For instance, that you can see here, We can insert genes into plants, get them to produce desirable characteristics. We can use them to for bioremediation or cleaning up, like waste spills and oil spills in land fills and in mines and that sort of a thing. We can use it to in various forms in medicine. So you can see here to produce human growth hormone, human insulin, attack blood clots, etc. So there's a wide variety of ways in which these things are used, and we're learning more about this just really in the news every day. Okay. So one of the things that's very useful for molecular biology is these restriction enzymes. And what these things are is basically like a molecular set of scissors. And what they're going to do, they're an enzyme that's going to cut DNA at a specific site. And if you know the actual reading frame that you want to cut, for instance, this restriction site here is GAA TTC, and it reads the same way in both directions. So it's referred to as a palindrome. And so what you've got here is once you've cut it that way, you can treat the other organism with the same type of restriction enzyme, and you'll get it to cut the same exact way. So then what happens is when you mix these two things together, you can see that they are going to line up, some of them, and that's why you have to select for them, because it doesn't work in all of them. It's only going to work in some, so you got to find a way to test for the presence of this. And then you mix a little DNA ligase in here that's going to form the phosphodiester bonds and link these little nicks together. And then once you've done that, now you've got this recombinant piece of DNA or this recombinant plasmid, and you can insert that into the bacterium and start to do your experiments with it. Okay, so main points here: restriction enzymes, they cut at specific spots called restriction sites. They look for particular sequences of DNA. Here's an example of one. As long as you know the site, you can call up a biotech company and you can get the restriction enzyme that you need. These things are found naturally in bacterial cells because what they're going to do is they're going to cut foreign DNA up to try to prevent it. For instance, like if a virus attacks the particular cell, it's going to inject some DNA or RNA in there. So these restriction enzymes are going to work to cut it up before it can take over the cell and kill it. Okay, so these things are found in nature, but they can also be manufactured by biotech companies. All right, what we said is they typically recognize nucleotide sequences that are about 4 to 6 base pairs in length, and these things are scattered throughout the genome. And typically you know what you want to cut, so you just search for the particular cut site that, or you search for the gene, you find the particular cut site that you're looking for, and then you get the restriction enzyme to kind of do your experiment with. Okay, it sounds really easy, it takes some time, but it's gotten a lot easier than in the early days. So Herbert Boyer, which we'll learn about, took him over 10 years to kind of learn how to do all of this stuff, but he became a billionaire overnight when he figured out how to insert the insulin gene into a bacterium and get the E. coli cell to start producing insulin for diabetics. So it certainly had its payoff, but it sounds easy, we're learning about it, but it does definitely take some dexterity and patience on the part of the researcher. Okay, so now one of the benefits now to these restriction enzymes is that they reproduce these cuts in a predictable way. So if it looks for GAA TTC, it's always going to cut at that particular site and nowhere else. So they're very specific and very reproducible. You just have to know what you're looking at. The key is now with these things is that they produce sticky ends. So in other words, we know that the DNA molecule is joined together by these various hydrogen bonds. Okay, so if we break it like this, if we cut it right here, what we end up with are two what are referred to as blunt ends. So these things don't really serve any useful purpose because it's really hard to get them to go together. Whereas when you have what's known as sticky ends, this thing will cut in a particular way where it would look like this. And now you can see there's spots here where we can insert a particular gene, and because they're going to kind of overlap, and those sticky ends will kind of bind together this way versus the blunt ends that don't want to bind together and line up and so forth. So what these fragments will typically do, some of them at least will hydrogen bond, then you can select for them. All right, so here's a movie based on how restriction enzymes work. I know that this link actually works too, so take a look at this and the other movie and then go on to part 2 here. Okay, so if you have any questions, make sure you write those down too, and we'll talk to you soon.